Okay, our next section is C21 chemical signaling, and this one is HL only as well. Alright, this one is directly related to the gene expression one. You're going to notice a lot of overlap between these two. Alright, so there we go. Standard one is nice and easy. It's our definition of a receptor. So a receptor is a protein that has a binding site for signaling chemicals known as ligands. Excuse me. So things that bind to them are called ligands. Okay. We're going to do standard 2, even though it wouldn't make sense to do it in this order, but that's fine. So standard 2 is all about bioluminescence. And so what you have here is what's called quorum sensing. So this is a type of sensing found in bacteria only. And so what they do is bacteria release signaling molecules called autoinducers. Autoinducers. Okay, so they release these signaling molecules into the environment. Into the abiotic environment. Right, so the bacteria, they're single-celled organisms. And so what it does is it triggers a cell response at a high enough threshold. The most common one it triggers, at least the one you need to know, is Vibrio fischeri. It's a type of bacteria. Technically it should be underground. I don't like that. And what it does is this quorum sensing it is what causes luminescence or bioluminescence. Biolumic. Oh, it's a long word. Bioluminescence. There we go. So bioluminescence, it causes it to glow, to produce light. And this only happens when it reaches that threshold, as the bacteria are releasing these autoinducers in certain environmental conditions, which then can cause it to do that. It does relate to other things. Works for other things as well too. That's the main one. Right. Standard 3. This is all about the types of ligands. And so there are 4 main categories, and then inside of those we're going to do standard 4 as well too. So there's hormones, and then inside of hormones you have your different ones as well too. So you have amines, which are modified amino acids. You have your proteins, which are proteins, and you have your steroids, which our steroids fall under our lipid hormones. So they can transmit differently and they bind to different receptors. For your neurotransmitters, you have another wide variety of molecules. Your neurotransmitters, we can have amino acids, peptides, so short proteins. You can also have amines or modified amino acids here as well. and nitrous oxide can also serve as a neurotransmitter. All right, so those are all different ones. They're going to react differently with different receptors. They travel differently, right? So these two are very different, and we'll talk about that in Standard 5 in a second here. But there are two more categories of ones left. There are cytokines, which are primarily for the immune response and like growth of cells. So cytokines are responsible for the immune response and growth, whereas your last one is calcium. And calcium is primarily going to be used by the muscles as a signaling molecule as well as neurons. And so those are the four main, it's calcium ions. Calcium ions. So Ca2+. And so it's primarily going to be in the muscles. Now, for our hormones, these are going to be long, this is Standard 5, these are going to be long lasting, and they are going to be distant effects as well as local, but they can go all over. They typically travel in the blood, occasionally in the lymph system, and so they can be distributed all over the body. And so these are through the blood system, whereas neurotransmitters are just between neurons, so they're very local and they're very short lasting. So they are much more temporary. So this right here, right, is our long, this is our short signal. And so both of these are often activated at the same time, this one to get a quick response and this one to keep a response going. So for example, in your fight or flight, right, you have both of these working together to help you run away from a polar bear. All right. Now, there are two different types of receptors, two main categories, excuse me, is a better way of phrasing that. There are our transmembrane receptors, and we have our intracellular receptors. So these are found in the membrane. The transmembrane ones are found across the membrane, right? Trans meaning through the membrane in this case. And so this one, the signal is going to remain outside, whereas on this one is inside the membrane, and its signal on the inside. Typically, the receptor for this one is often like non-polar receptors, whereas this one, it's typically receiving polar signaling molecules. And so for this one here, if we remember, like this part is non-polar, but the part sticking up here is typically polar. Whereas for this one, it's like this, and so this part is often non-polar, where it interacts. And so if we look at our list above, right, so most of our neurotransmitters are going to react with transmembrane receptors because they're polar, whereas our steroid hormones especially will often come inside the cell and interact with the intracellular receptors. Okay. All right, and so that is our types of receptors. Then what we have is what actually happens. And so when a receptor receives a signaling chemical called a ligand, we get what's called signal transduction. And so what this means is it's starting a series of responses, or like a cascade effect or a series of responses. So it binds to a receptor, and then the receptor goes, whoa, and a bunch of different things happen. And now what we're going to do is go through the different things that can have happen. So the first thing that can happen is our neurotransmitter example. And so our main neurotransmitter is acetylcholine. And so acetylcholine, part of it is coming, the acetate is coming from the Krebs cycle, from the link reaction, and then the choline is coming from just being recycled. And so what you have happen is this neurotransmitter is released from the presynaptic neuron, and it goes to the postsynaptic neuron, where it binds to a receptor on the membrane. So this is going to be a transmembrane receptor, and then that receptor will then open sodium ion channels this opening of the sodium ion channels will start an action potential. And then acetylcholine is actually going to be broken down and sent back to the neuron. Okay, so this one is all about causing action potential. So an action potential is your neuron, the process of a signal going down a neuron. And so what this does is it changes the voltage, which then causes an action potential, which causes a signal. Okay. It can also signal the muscles and then cause muscle contraction too. All right, the next big category are our G protein receptors. So our G protein receptor is found in the membrane. It's called a G protein coupled receptor. 1, 2, 3, 4, 5. 1, 2, 3, 6. Yeah. Oops. Yeah, that's right. 6. And then you have the G proteins. So these right here are the G proteins. This is the G protein coupled receptor. The G protein coupled receptor. And it will bind something here, and it will trigger and activate the G proteins. The G proteins most of the time have GDP, and then when they're in the active form, they have GTP. And so there's alpha, beta, and gamma. That's a really bad gamma. That's a gamma. Just use your imagination. That's a gamma. All right, and so what these do when they're in their active form is they go cause other effects. Lots of other things. One of the things that they can do is, a specific example is when adrenaline or epinephrine, they're the same thing, when it binds to the GPCR, it triggers the G proteins, G proteins, which then trigger CAMP, or cyclic AMP. Cyclic AMP is a secondary messenger, and then that will then go cause other changes, and it leads to changes in expression, and it's going to be changes in like heart rate, and also changes in glucose absorption. So it's your fight or flight response, right? So, fight or flight response. Okay, so that's your adrenaline or your epinephrine receptor. It activates the G protein coupled receptor, which activates the G proteins, which activate CAMP, which then changes your heart rate, causes you to basically run faster. Okay, the next one is insulin. So insulin is the ligand, and insulin is going to bind to an insulin receptor. And that insulin receptor looks kind of like this, right? So we have this part here. Here's the membrane. So insulin will bind to the receptor, and what that will do is activate this part of the domain, which is a tyrosine kinase. And a tyrosine kinase is going to take this and add a phosphate to a tyrosine. So it's going to phosphorylate a tyrosine using an ATP. Okay. And then this is going to go affect gene expression as a transcription factor, which is going to cause more GLUT4 to be made, which is going to put the GLUT4 into the membrane, and it's going to cause glucose to enter. And so, so insulin binds, tyrosine kinase phosphorylates a tyrosine, it activates this protein, which then goes and acts as a transcription factor to affect gene expression. There's some other steps in between you don't need to know. This causes the upregulation of GLUT4, which is a glucose channel, which then causes that to be added to the membrane. Remember, ribosome, REF-ER, Golgi apparatus, membrane, glucose channel is added, and glucose will enter the cell. Okay, so that is insulin. So that one is a transmembrane. So all of those examples were transmembrane receptors. Now we're going to look at some intracellular receptors. And then we're going to start with standard 12. And the main three we need to know for this one are oestradiol, which is another word for estrogen, progesterone, and testosterone. These three are all steroid hormones. They're all steroids, and they all bind intracellular receptors, which then activate a pathway, which then causes transcription factors to be activated, which are going to increase gene expression. Okay, so all three of these are about increasing gene expression. Questions? Okay, so then standard 13 is, well, what do these two specifically do? And so estradiol acts on the hypothalamus, and it causes an increase in GnRH, which is the gonadotrophin-releasing hormone, which causes FSH and LH to be released, which leads to the menstrual cycle. But specifically, the first part of the menstrual cycle leading to ovulation. So not menstruation, the other half, which we'll know about more in a second. And then progesterone acts on the endometrium, and it prepares it for pregnancy. Prepares it for pregnancy. And remember, the endometrium is the outer lining of the uterus. So it causes the thickening of the outer lining of the uterus, and that's standard 13. All right, standard 14. The cell pathways, right, have positive and negative feedback loop. And so positive feedback is, as it increases, it causes more to be made, whereas negative feedback causes it to be suppressed. And so our example for positive feedback was the cyclins. So as you create more cyclins, this creates more cyclins, which creates more. Remember our graph that looks like this in our mitosis section. Whereas our negative feedback example is testosterone. And so as you increase testosterone, it actually will act on the hypothalamus, and it'll cause it to be down-regulated until it goes back down again. So increase in testosterone actually will signal and cause it to stop secreting more testosterone, because you only need so much of it at a time, and so this causes you to not create too much testosterone. And that is chemical signaling.